to hear 10 to 11 is all it is.
Right.
So they should be able to hear you now, but you only have one viewer.
This is so frustrating. Okay.
Because for some reason you have copyrighted information.
No, the YouTube please let me see.
Oh, wait. Yeah.
Try the 10 o'clock one.
C word.
Corona.
Oh. Right.
No. This girl said that YouTube shut down anything about Corona because they don't want us just spreading
Misinformation.
Fart. Right.
Oh, alright.
The current viewer is probably Connor.
Yeah. Actually, it's not. I don't think.
Okay. Hey guys. We're gonna use the C word instead of the actual CRO beer word. So maybe we'll just call it beer. Just an idea. Oh, the 10 o'clock is just me trying to figure it out.
No, go to the 10 o'clock live one.
Go to 10
There should be a live.
live.
10 to 11, that's like 23 minutes. And then there's a 10 to 11 that's live.
Not the 10 to 11.
I can.
Go here.
No, I need to go to here. And
Delete it.
delete it. No, not there. Go back.
Oh, this one's live.
Videos.
We're going to share that link now. Share a new link so they can see it.
There's that one. There's that one. That's the 10 to
Did he
11
just
one.
ride his bicycle through our yard?
I have no idea.
Surely he was in a hurry.
And just hit.
Trash.
Hey, you got one like on it though.
Yeah.
Oh, I can do this.
Delete forever.
Delete forever. I understand deleting is permanent. I don't need that one right now. Alright, so you're still live.
Alright.
You should.
Can you copy that link and I'll post it in Classroom right now.
Oh, it's studio.
Share. Should be a share option somewhere.
No, you don't need this. Because you're still technically live. So you need to find where. Dismiss. Don't care. Blah, 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 blah. Video analytics. Yeah, with this one, right here. Okay, so you're back to here.
Alright.
They should see that. Alright.
Alright.
You have 6 viewers now. Okay, so now, boom,
Boom and boom.
and boom, but then you need to go to here, and then, I think it's this chat, or is it that chat? I think it's this one. Actually, it's neither one of those. Okay. pop this out real quick. Go to there. Make it smaller. This one, go to there. All right. So that's what they're seeing.
All right. Good night. Hi, guys. So, I guess the C word really is a bad word. Hopefully, we'll get to we'll get a little further than we did last time. If not, we'll just use old school, and I'll make a video on the desktop, and I'll share it through Classroom the old-fashioned way. It's quite frustrating. I feel like I'm in Fahrenheit 451. What?
Zoom in and be focused. A little hard to see.
Okay.
Yes. If you can zoom in to make the letters bigger.
Okay, I'll hurry, Rainbow Kit. And like I said, we'll figure this out. This is ridiculous. So I'll try to teach this without the C word. All right, so hopefully we got here where viruses have to attach, insert, they have to replicate, and this requires the creation of genes. This requires the activation of genes. And that activation of genes will create DNA polymerase or RNA polymerase. And we know that the C word doesn't need that because it enters the cell ready to go. So it's ready to assemble and do protein synthesis. And it's going to synthesize particle parts for the virus. And
You just said protein synthesis, so that's your buffer time.
okay. Assemble parts through protein synthesis, and then it's also going to synthesize the enzyme, the N enzyme, the neuraminidase enzyme, to allow the cell to allow the viruses to pop the cell, to lyse the cell, and release those new particles. And this is where the infection part gets enormous. So natural immune response, you've got the histamines, which is a chemical signal. That's that ligand that's going to bind to receptors in your blood vessels, cause the blood vessels to dilate, and when the blood vessels dilate, the plasma in your blood can leak out. White blood cells, antibodies can all leak out. And that's great because those antibodies can disable a viral particle. They can disable pollen particles. They can disable germs before they replicate and attach to your cells. Then there's the inflammation. Inflammation is huge because it's just very general. It's again, it's a lot of it's because of the histamines released, but the tissue swells. It gets bigger. The blood flow to that area increases. So the area is red, it's hot, it's swollen, all because of the extra blood vessels. So if you've got a stuffy nose and a runny nose, that's because of this inflammation that's happening. It's just a natural immune response to protect your body. if you twisted your ankle or broken your toe, that inflammation helps because it stabilizes that bone. Then we have the cytokines. Cytokines are additional chemical signals that initiate other immune responses. They can increase inflammation, as we see with the C word, and they can initiate fever. They activate things we call cytotoxic cells, which kill viruses. And any cell that has been infected by a virus, these cytotoxic T cells attach to and just blow them up. So it's like amazing power. So like Midas had the touch where he touched stuff and it turned to gold. Well, these cytotoxic T cells touch a cell that's been infected by a virus and kill it. Pretty amazing. Now, with this immune response, we do have conditions, autoimmune diseases, autoimmune diseases like lupus and rheumatoid arthritis, where the immune system of the person attacks its own cells. Ms. Dalton and a couple of other teachers have rheumatoid arthritis, and they are always on immunosuppressant drugs to tell their bodies to chill out, because their body is always about this inflammation and always about this cytokine storm, which you may have heard on the news. So, some of the medications you've heard about recently to treat the C word have been used not only for malaria. See, the media only cues you into a little piece of that information. See, when you have a studio full of producers, you can have people to do that kind of stuff for you. All right, so where was I? Rheumatoid arthritis, cytokines. Oh, the same drugs that treat malaria also have been used and are currently being used for lupus and rheumatoid arthritis patients. And with the recent information about hydrochloroquine, and people with lupus and rheumatoid arthritis are having issues getting their medication because some people upstream are collecting and maybe hoarding the information. If you are on immunosuppressants but get infected by a disease, you will have trouble fighting it. It's going to take you a lot longer to fight it off, and you're going to need some additional help. The physician won't want to take you off or lower the dose of the immunosuppressants. You're just going to be sicker longer, and you're going to suffer, which really stinks. Good question. Okay. Soothe. Sorry for the uhs. Okay, so this particular virus, the C word, and flu viruses are the same. We classify them in the same realm, like you had kingdoms and phylums and all that mess. Well, with viruses, they have realms. but they're in different phyla, order, family, all that other mess. So they're kind of the same in the fact that they're viruses, and they're kind of the same because they're RNA, but they are different in every other aspect. Both of these types have pieces of RNA, and they are protected by a lipid bilayer. Didn't realize that, we never emphasized that before, but this virus is contained in a phospholipid bilayer, just like the rest of your cells. Where do the viruses get that phospholipid bilayer? Just like endosymbiosis. They caught it from when they ejected themselves from the host cell. Embedded within that phospholipid bilayer are proteins and glycoproteins and glycolipids. When were those made? Those were made up here during the assembly parts. So the host cell made those viral glycoproteins, glycolipids for the virus. Now, some viruses have an extra capsid, and that extra capsid is made of protein. So I'm kind of jumping ahead, but why does washing your hands with warm, soapy water kill the virus? Let me repeat the question. Why does washing your hands with warm, soapy water kill the virus? The soap is an important part, and the warm is an important part. Okay, hopefully you got the fact that the soap, being slightly pH basic, is going to dissolve the phospholipid bilayer, and the warmth is going to... Good, Julie, awesome. Excellent. Yes. The soap, the basic, the alkaline soap is going to dissolve that phospholipid bilayer, just like it dissolves the greasy fat out of the lasagna pan. Now, what about the warmth? What about the heat? How is that going to disable the virus? Hopefully, you gathered that the heat or the warmth is going to disable the virus by denaturing the proteins in the membrane, so that even if the virus survives, the protein receptors that are supposed to bind with the host cell have mutated, or not mutated, mutated is not the right word. They have what's the word? Denatured. Good job, Bianca, that's 2 and 2. Good for you. So you're denaturing not only the proteins on the membrane, but hopefully you're denaturing and causing the DNA or the RNA inside the virus to fall apart, which is a win-win. So don't hold your hands under like boiling, scalding water. A little warmth is enough to denature the proteins and to denature the DNA. Honestly, the warm soapy water is enough to kill most microbes. Now, the hand sanitizer, alcohol has an awesome way of dissolving fats also, because it has a polar end and a non-polar end. And so hand sanitizer is a great plus if you can handle it. And those burps were for our potty break. That's a joke, haha. Okay, now the flu virus, as I mentioned maybe earlier, you got it, is a negative RNA virus. So it's going to come in and it has to do some transcription. So it's going to come into this process. Where's my process? It's going to come into the process here. DNA viruses come in here. What about the toilet paper? You know, I think the toilet paper bit has something to do with there's a new way to make meth, and instead of using Sudafed and other over-the-counter drugs, they're using toilet paper. So you used to have to show your driver's license to buy cold meds. Now you're going to have to show your driver's license to buy toilet paper, so they know you're not making meth out of it. Just a joke, Sarah. Good job. All right, so DNA viruses with DNA have to jump in here, and they've got to do replication first. They've got to copy their DNA. You know you miss it, Goodwin. And then they're going to transcribe that DNA to mRNA, and then they are going to translate it. Negative single-strand RNA, like the flu, jump in right here. So they can replicate much faster and therefore spread faster. The C-word virus jumps in here. It doesn't need to mess with this. So what is it doing during that latent period? It's doing this very, very quickly and very, very efficiently. So it can do this without triggering an immune response, without triggering our white blood cells and our memory cells, and cause all kind of inflammation and spread before you exhibit any symptoms. Sneaky, 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 huh? Okay, so yeah, so that's what we're talking about. So this C-word virus and replication method allows it to be more efficient and more dangerous than the influenza. All right, so a couple of you asked the question, and I had the same question too. Influenza, the flu virus, is different than the C virus. Okay, now the video you watched did a very good job of explaining the hemagglutinin gene that allows the viruses in, and the neuraminidase gene that allows the virus out. And so these genes are going to code for a protein that allows those viruses out. And then this gene codes for a protein, specifically an enzyme, that makes the enzyme that allows the virus to lyse. The C virus does make or have similar genes as do all viruses. They just simply aren't identified as H and N's. I don't know if that makes any sense. So the corona, dadgummit. Oh, good question, Julie. You're jumping again. Awesome. If the virus can't leave the host cell, then there's no more infection, is there? And the virus doesn't get spread. And there are, as your genius self may have predicted, there are antivirals that do exactly that. They prevent the lysing of the host cell. Good job. And I have that name written here somewhere, the name of the virus that does that. But, the name of the drug that does that. I don't suppose it matters. All right, so yes, good job, Julie. You just created $1 million for yourself and the pharmacological industry. All right, just an interesting fact the phospholipid of the virus, of the flu virus, 80% of the phospholipid proteins, I'm sorry, let me rephrase that. 80% of the proteins in the membrane of the virus are H genes. I'm sorry, they are H proteins. Only 20% of the proteins in the membrane are N proteins, which kind of makes sense. So in the membrane, there are more keys. Yes, it does. The warm water can denature the protein capsid, absolutely, causing it to fall apart. And as you might guess, viruses with a protein capsid are a little more challenging to kill or to disable than are viruses without a protein capsid. Okay, so back to the phospholipid bilayer. The influenza viruses have more keys to get into the cell than they do ways to get out, but that's okay. They will generate these proteins once they're already inside the cell. All right, so I asked this question. We talked about hemagglutinin genes that make the enzyme that allows us into the cell, but hemagglutinin esterase genes. Esterase is the key. Do you recall talking about acetylcholine esterase? We know that acetylcholine initiates the nerve signal. Acetylcholine esterase breaks down acetylcholine. So I was hoping you would jump to the idea that hemagglutinin esterase is the enzyme that allows that original virus particle to detach from the host cell. I hope that you gather that an esterase is going to reverse any action. Okay, a lot of you picked up on this, and I gathered that from the comments you made about the video. I don't want to mess things up. Okay, all of these are dangerous to patients who have compromised lungs. But what the media is not telling you is that they are also extremely deadly and infectious to people with strong, healthy immune systems. Now, I want to talk to you about this right here. If you've watched it all, you see the data. How many old people are being infected? How many firemen have been infected? How many policemen have been infected? How many doctors? Right now, in Texas in particular, and in the United States, who are we testing for the C? We are testing the old, and we're testing those right on the front lines, which is logical. So when we get the data, it makes sense that we're going to see higher numbers of old patients with it and firemen, policemen, medical professionals. We aren't testing the young. We aren't testing people my age. We're not testing people your age. We're just saying if you've got the symptoms, stay by yourself and quarantine. So the data that we're seeing is skewed. Also, media and reporters aren't listening to the whole story. They are jumping to the conclusion and saying, oh, you're immunocompromised, you're going to die. Well, yeah, that's true, but you're also at risk if you have a strong, healthy immune system. Because people with a strong, healthy immune system are going to do a very good job of developing inflammation. They're going to do a really good job of swelling up those lungs, where the virus is, because that's what our body does. When you get a rhinovirus in your nose swells. Yes, you're right. Celebrities. Just tell us, all those science guys who never made it through 9th grade biology, and they're going to tell us all about it. Good comment, Yusuf. Anybody with a strong immune system is going to make lots of this and lots of this. And that's creating the inflammation and the difficulty breathing, which ends up killing our patients and causing them, before they die, they have to be intubated. They have to be hospitalized to be intubated. And intubated, for those of you who don't know, that's when they put the tube down your trachea and force oxygen into your body because your lungs can't do it. You can imagine it's quite uncomfortable. So, rambling on about having a good immune system, will the person survive? Yes, absolutely. But they're going to have trouble, and the likelihood that they may be young and have a strong immune system and have to be hospitalized after all is a possibility. What is the body doing while it's inflamed, and what are these cytokines trying to do? They're trying to give the body time to make B cells and T cells. And the B cells are going to make antibodies that stop the virus, and the T cells are going to make cytotoxic T cells that are going to touch kill the virus. What else? Oh, and also then there are memory cells. Now with older people, as with the Spanish flu, those older people, they probably have memory cells. Memory B cells and memory T cells deep in their body, ready to go and fight the new virus. But because they have COPD, emphysema, tuberculosis, the inflammation involved is going to hurt them. I don't know if you have any comments or questions about this. I'm going to stay hushed for a second to give you time to think about the strong and healthy immune systems being dangerous or deadly. I see we're over the 104 time limit. We're almost done. Can we hang in for a little bit? All right. If you gotta go, I get it. Okay, I feel like I'm rambling and I can't see your face, so I can't judge if you're getting it or not getting it, or if I'm repeating myself too much, but anyway. Like that and so forth and such as. Okay, so this is just some random data that I found on various different sites. The Spanish flu killed this many people, and this was probably in a 2-year period. Hong Kong flu was probably 1 million in probably a year period, 2-year period. SARS only had this many deaths. It was a really short infection virus. And the avian flu was transmitted from bird to bird, bird to human, but fortunately this N, and I think Sarah, you asked a question about the N. The N was not changed or did not mutate, fortunately, so it wasn't human to human, unless Tumpy gets itself involved and changes it. Swine flu, Kat, I think, is this the one that your mom contracted? And bless her heart, thank goodness she didn't pass away. This number, that looks high. I wonder if that's an infection and not death. All right, MERS, which was called COVID-12, which is similar to the C one, but it was COVID-12, not 19, because it was in 2012. This was a very long time period, and it had only this many deaths. So, and then Ebola, this was a short epidemic, and had this many. And then there's here, I did include a live link if you're interested. This was when I first created this document. This has to be infections. This can't be deaths. And it's global. I don't know. I can't remember. That's why I should do a better job of documenting my work. All right, so what my point is here, data. Data is only reliable as the source, and it's only reliable as what's being reported. If you don't take the test, you're not going to fail or pass it. So keep that in mind when you're seeing the data. Also, when I looked up data for the HIV infections, like Brazilians of people, lots of people have died from HIV, but the data I saw was like from 1984 to present. So when you think about that time period, you're expected to see a lot more deaths. So when you're looking at data, consider the source, consider what the data is based on, who's getting the test, who's not, and the time period. How long is the data collected for? All right, so this is just out of your book. Hopefully you looked at it, you read at it. This positive sense information is what I've kind of driven home the entire time. All right, so we're going to jump into treatments. Treatments. All right, so the first treatment that you may have heard of online is hydrochloroquine and this antibiotic. And you've been told over and over and over, antibiotics don't help with viruses. Antibiotics don't help for viruses. Well, it depends. Now, let's look why this treatment might be successful. Hydrochloroquine reduces inflammation. We've talked about that. That's why people with lupus and rheumatoid arthritis and malaria take it. It's reducing the inflammation. The antibiotic azithromycin, which is the Z-Pak we all took when we had a snotty ear infection back when. This antibiotic stops protein synthesis. Okay, so why would this work very well to stop any virus for that matter? Let me repeat the question. Why would this, theoretically, this is not approved by the FDA, this is anecdotal only, it has not been studied, it has not been done anything. Why would the combination of medications, hydrochloroquine and Z-Pak, actually theoretically help treat C virus infection? Okay, so hopefully you can figure it out that this deals with the inflammation deep in the lungs. It's going to allow the person's lungs not to fill up with fluid. And then this is going to prevent the virus from synthesizing the protein it needs to escape. So if it can't synthesize its protein particles, it's not even going to reach the point of being released So basically, you're stopping this part of the viral infection. Or on my pretty page, where's my pretty page? The Z-Pak stops this part. And so it's just like a signal transduction pathway. If you can't do this, you don't get the end result. So we're blocking the pathway, and so we stop the virus with the combination of the two. Now, again, this is anecdotal information. It has not been tested. And this hydrochloroquine has a lot of side effects. It's not good for, awesome, you guys are awesome. Good job. Awesome, awesome, awesome. Bianca, Yusuf, Hoops, good job. And I'm sure the rest of you are hopefully the rest of you are getting it. You're just not commenting, or either you've left because you've got other stuff to do. So you can't just give medication just to give medication. This has really bad side effects. It causes issues with the person's corneas. Vision's kind of important. That was sarcastic. It also causes issues with heart, blood vessels, blood pressure, and we also don't know what it does long term on the structure of the blood vessel itself. So it's just a temporary fix. Is it going to cure them? No, they're still going to suffer. It's just going to make the side effects a little more bearable and maybe shorten the infection. All right, there's another this is an antiviral, I can't even say it, remdesivir, and it has been used on the Marburg virus and the Ebola virus, which are both negative SS single-stranded RNA viruses. So they are very similar to they're very similar in function to influenza. So remember, negative single-strand RNA inhibits RNA polymerase. So it's going to inhibit the making of RNA. Now, it's still going to allow for this transcription and the translation, but what it's going to slow down is to make more virus particles. Oh, let me hush. This medication does this. It inhibits RNA polymerase. Remember, RNA polymerase is necessary for making more RNA. In order for the pathway to get to this point, these viruses have to have an opportunity to make RNA to go into all of these little particle parts. So this medication might help because it's going to prevent the making of this. It's not going to prevent the creation of RNA polymerase. It's not going to stop the protein synthesis of all the viral particle parts, but hopefully it will stop the actual copying of the virus. So you basically, what you're going to get is a bunch of shells, but no RNA inside, hopefully. Again, this is being, this particular medication, it's been approved by the FDA to treat these two drugs, or these two diseases. It has been fast-tracked to treat the current C-word virus. We'll see. Again, we have anecdotal evidence. We don't have concrete, repeated, successful trials. All right. Vitamin D is an awesome bit. Just eating foods high in vitamin D, but more importantly, getting out in the sun, because your body really does make enough inactive vitamin D You just have to be exposed to the sun to activate it. So take off your shirts, put on that sports bra, put on that tank top, put on those shorts, and get some sun. Okay? Maybe simply being out of school and available to be in the sun will save the lives of many people. Vitamin C. Vitamin C does a good job of cleaning up our bodies after everything is said and done. So as the body fights and disables germs and viruses, it's got to have a cleanup crew, and the vitamin C helps do that. All right, what we talked about soap and warm water. What about UV light? Okay, you know that UV light, I just told you to go out and get some sun, but I did not say get sunburned. UV light does burn our skin because the powerful UV light penetrates and damages the skin and the tissues below. UV light damages and kills germs. If you've ever I guess we can't go into a store, but if you've gone to a restaurant, I know that I saw one at Brew and Bake, they had one at Firehouse Subs, and they have one at HEB as you walk out the door kind of by the pharmacy, by the self-checkout. If you look up, there's this white tube kind of looking thing. I'm afraid to change what I'm showing you. What that is a UV light, and it emits UVC light. And it has a little fan that pulls in air, and it exchanges 600 cubic feet of air per minute. So it's sucking air pretty fast, and as it pulls that air through, it exposes that air and anything in that air to not UVA, not UVB, but UVC. UVC is a less powerful UV light, but it kills germs. We already have it in place in food establishments everywhere. I think Miss Richardson in Sea Wing, who does the culinary classes, has one. It's a very common, it's been used forever. You don't even notice it because Vienna is so quiet. It's naturally sterilizing. There is a possibility, and no one in the media is talking about it, but there is a possibility that the presence of this will naturally sterilize a very large space, a very large room, all at one time. So that's hopeful. Okay, let me let you process that for a second. I'm looking for test. I'm looking for my notes on how to test for COVID-19. If you need to go, I totally get it. But if you can hang on, we're going to cover testing for COVID-19. Oh, maybe not. I think I'll post the Carson, are you here? Okay, if you can stick around, we'll go ahead and try to do the testing bit. I can't find my pretty notes on this. Well, boogers. Alright, we'll just have to do this rough. Alright, let's try this here, this. Oh, here, sorry. Alright, I can't find the chat stream. Alright. Okay, so we are on testing for C. I can't find my pretty notes on this. I really did make nice, pretty, readable notes, but yeah, you can guess that my kitchen table, my dining room table looks like my desk right now. Don't judge. Okay, so to test for this, we do a process called reverse transcript PCR. The first thing you need to know is PCR is polymerase. I'll make these notes nice and pretty for you and share them with you. Chain reaction. And I'm going to try to show you a video on PCR. So if this doesn't come through, somebody message and tell me that it's not working. This explains the general process of PCR. Okay.
The purpose of PCR is to create copies of a specific region of DNA. To use this technique, researchers must know the base sequences at either end of the region of interest. They use this information to create primers, short sequences of single-stranded DNA that have complementary sequences. Researchers mix together the DNA to be amplified, the primers, a DNA polymerase that retains its activity at high temperatures, and free nucleotides. Click the Up button to increase the temperature and begin the process.
Oh, sorry. So I'm going to heat it up.
The mixture is heated to 92 to 94 degrees centigrade to separate the strands of DNA to be amplified. Now click the down button to lower the temperature to 50 degrees so the primers can base pair to the template strands of DNA. Click to raise the temperature to 72 degrees. At this temperature, the bacterial DNA polymerase uses the primers as starting points to synthesize complementary strands of DNA. This completes one round of PCR. The number of copies has been doubled. Click a temperature adjustment button to begin the next round of PCR.
Okay, so we're gonna heat it up. By heating it up, you're breaking.
Click to change the temperature so the primers can base pair with the template DNA.
By heating it up, you're breaking these hydrogen bonds between the A's and the T's and the G's and the C's. You pull it down.
Click to adjust the temperature so that the DNA polymerase can synthesize complementary strands.
So now the primer, the starter, it's like a capital letter at the beginning of a sentence, bonds, and now we're going to warm it up a little bit.
This second round of PCR has doubled the number of DNA molecules again. Repeated cycles of PCR can quickly create millions of copies of a DNA sequence in only a few hours.
Okay, so this really, we used to do this by hand, and when I say used to, this process wasn't really made public until 1996-ish. We didn't start even thinking about teaching it in AP Biology until 2004. At that time we did all this by hand. We had a hot water bath and a cold water bath and a warm water bath, and we would just move it, and it was very time consuming. Now they have machines. They used to be $10,000. Now they're $500, but they would do this exact process. Now my question for you is, why would you need to do PCR? Back up. I'm telling myself to back up. Polymerase chain reaction. We're copying the DNA. We take a tiny, tiny bit of DNA and we make tons and tons and tons of copies. And you can do this in 3 hours. Done. Okay? So when they heat it up, they chill it and prime it, and then they heat it up a little bit so that they can make the copy of the DNA. And they're using a DNA polymerase from an extremophile, a thermoacidophile archaeobacteria, because it's an enzyme and it works at very warm temperatures, and it works when the DNA splits, and any other protein would be denatured, but this particular thermoacidophile DNA polymerase does not denature as at high temperature. So we're kind of stealing or borrowing from the weird Mother Nature that is our archaeobacteria to do this process. All right, so why would we need to take a tiny bit of DNA and amplify it? Why would we need all these extra copies? One place this is used is in forensics. We can take a hair follicle, we can take the cells from the bottom of that hair follicle, take a tiny piece of DNA, amplify it, and do a Brazilian test on it so that we can prove that Billy was not at the scene of the crime. It was actually Sam that was at the scene of the crime. We use teeny tiny bits of DNA. If you've Ancestry.com or 23andMe, you just take a little swab of your DNA, you send it off to the company. They're going to do the process of PCR, and then they're going to run all those copies or all those tests on that amplified section of the PCR. We can test for parental, who's the mom, who's the dad, kind of thing. We take a little bit of DNA, we amplify it through PCR, then we do a process called gel electrophoresis, which separates it, but we'll talk about that anyway later. So that is PCR. So with this particular virus, first we have to do something called reverse transcription PCR, because if you remember, this virus is an RNA virus. So So we have to take the RNA, turn it into DNA, and then do PCR. Look at that alphabet soup there. There is an enzyme to do this. That enzyme is called reverse transcriptase, because transcription goes from DNA to RNA. If we're going to go backwards, which is what we have to do with any RNA sample, we use this, we add the reverse transcriptase enzyme, and it gives us this. Then we can run PCR. So when we do a test for the C virus, we take the sample from the person's, deep in the person's throat, and we're taking just a small sample of their cells. We're going to isolate the RNA, because we only want RNA. We don't care if they've been exposed to DNA. We're going to turn it into DNA, and then we're going to amplify that DNA that matches the code or the pattern of the C virus. That test takes time. It takes about 3 hours, but it takes time to get that test to the company to run the test. It takes time for that person at that company to take the tubes out and run the test. And it takes time for that person who's running the test to take it back and report their findings. And that's where the lag time is. So do you have any questions right now about reversed transcript PCR? We're sampling the person's body. We're amplifying the RNA. And then we're identifying, do they have the RNA or not? Either I'm dead and the stream is ended, or everybody's left, or the people who comment don't have anything to say. Okay. Oh yeah, thank you. Thank you. All right, the next test is called the ELISA test or ELISA test, depends on how you want to say it. And this is a blood serum test. Basically, it's testing for antibodies. This is similar to what's done for the HIV test. It is very fast. Takes no time at all, and here is a demo of how ELISA works. This guy's boring, but he's good. I wouldn't share with you bad stuff. This is a quick little 1-minute video. So here we are, the enzyme-linked immunosorbent assay. Again, we're testing the blood to see if the blood possesses antibodies that we are looking for.
Sorbent assay, or ELISA, is a commonly used format for serologic testing. The purpose of this animation is to explain how this test works. ELISA serologies are usually done in multi-well microtiter plates so that dilutions of serum can be easily prepared and tested. To better understand how this assay is done, let's take a closer look at what happens in one of the wells of this assay plate. To perform the assay, the wells of the plate are coated with the antigen of interest. For commercial tests, this would be done by the manufacturer of the assay. To begin testing, the wells are filled with dilutions of the patient's serum. If antibodies against the antigen are present, they will bind to the antigen fixed to the bottom of the wells. But only antigen-specific antibodies will bind to the wells. The wells are then washed out to remove all unbound antibodies. Next, a solution of an animal antibody against human antibodies is added. This second antibody is covalently conjugated to an enzyme. The wells are washed again, this time to remove any unbound enzyme-conjugated antibody. Finally, a solution of a colorogenic enzyme substrate is added. The interaction of the substrate with the enzyme on the second antibody generates visible color. The development of color in the wells with a specific antibody can be seen with the naked eye or quantified with an electronic plate reader. The reaction is less intense as the serum is diluted and the amount of antibody captured in the wells decreases. The titer is the highest dilution with definite color development. Next, a solution of an animal antibody against human present. They will bind to the wells of the plate are coated with the antigen of interest.
Okay, so where or what would this antigen be? If we're testing for the C, where is this antigen? What is this antigen? Hopefully you said the antigen is probably going to be the external protein receptor on the C virus. Okay. This is what is going to be paired up with. So if there is any antibodies,
Antigen are present, they will bind to the antigen fixed to the bottom.
So you see, this is an antibody, but it's not the antibody we're looking for. It's not the droid we are looking for. We're only looking for these antibodies, so it doesn't bind.
Of the wells.
So it goes away.
But only antigen-specific antibodies will bind to the wells.
So this allows us only to find the antibodies for the particular virus, whether it's HIV or C, that we're looking for.
The wells are then washed out to remove all unbound antibodies. Next, a solution of an animal antibody against human antibodies is added. This second antibody is covalently conjugated.
Okay, so this is from an animal. You know, if I mix my blood with monkey blood, the monkey's antibodies are going to attack my blood, and probably my blood's going to attack theirs too. No animals were harmed in this experiment, but we're simply using this to identify where the human antibodies are.
Conjugated to an enzyme. The well.
And we have synthetically attached this fabulous little Pac-Man looking enzyme to the animal antibodies.
Wells are washed again, this time to remove any unbound enzyme conjugated antibody. Finally, a solution of a colorogenic enzyme substrate is added. The interaction of the substrate with the enzyme on the second antibody generates visible color.
Okay, so this enzyme binds to the active site attached to the animal antibody and changes color. Yay for color
The
changes.
development of color in the wells with a specific antibody can be seen with the naked eye or quantified with an electronic plate reader. The reaction is less intense as the serum is diluted.
So you would expect a patient with lots of antibodies would have a very, very dark color, and a person with very few antibodies is going to have a very light color. Hello, Ladybug. Okay, okay. Good girl. Okay, so let's try to go back here. Maybe. Okay. And I lost my chat. Okay, so that is ELISA. We're testing the blood for antibodies. Now, as I mentioned, ELISA is very fast, but it tests for antibodies. That doesn't necessarily mean that the patient has the disease right now, but it has had that disease in the past, at least long enough to create antibodies. Now, well, what's a problem with this test? As I just mentioned, you could not be sick, not even contagious, and still have the antibodies, but it could detect an active infection. However, a person could take the ELISA test, be infected, even being showing symptoms, but the body has not had time to make antibodies. That takes time to make those antibodies. So you're going to get a negative ELISA test, but you're definitely going to have the virus that you're looking for. So it takes a little while. That's why people with HIV and the C who do that test will get a false negative read. But it is an awesome test, and I'm going to put up a recorded video on how we can use that information later. Okay, and back to the reverse transcript PCR. Hopefully you would gather that a bad thing about this is the time and the handling and the errors you get with this particular type of test, because there's lots of room for error here. You could have, when the physician is taking the sample, did he get enough of the cells? When the person's sticking it down your throat, did they get the tube or get the swab back in the tube efficiently? Then there's the time factor. Was the virus still viable by the time it got to the company and got analyzed for the presence of the RNA we're looking for? So there's lots of room for human error in this. Also, if you're getting a test early or you don't get enough cell sample, you're not going to have enough to get it done. All right, guys, I'm going to stick around for a little bit longer. That wasn't as neat and polished as I hoped, but as I've said before, none of us have asked for this. I miss you guys. That was a 2-hour lecture. I'm sorry. Do you have questions, comments right this second? I'm going to stay quiet for a little bit and let you type. Okay, I'm gonna stay logged on for a little longer in case you have something to comment or share. If you want to private message me, that's great. If you can email me, that's fine. I'm gonna find some quiet time. It might be after midnight, after everybody goes to sleep. I'm gonna present a polished version of this chaos in briefer little bits. And I've been told that rumor has it that we will be downloading Google Meets to student Chromebooks. It's just a rumor, so we all know what rumors do. But other than that, thank you guys for participating. I hope it cleared some things up. If questions or ideas or comments come to your brain later, email me. It's all good. We're all here to learn. Again, I thank you for your patience and your understanding. Like I said, I'm going to leave the channel open a little longer for you to type, and if you're done, then you can feel free to leave. No big deal. Because I'm taking off these fancy little headphones. Yep, I'm taking these fancy little light up headphones off, and I'm gonna walk away and allow you guys to chat or comment if you need to. And other than that, thanks guys. Peace out. And stream.